Yeah.
Okay, so very excited today to have with us Dr. Jia Zhao from the University of British Columbia. Title of her talk is Stem Cell-Derived Islet Models Generated with a Tunable Differentiation Strategy. A little bit about her. Dr. Zhao received her doctoral degree from Peking University, where she studied islet development and beta cell function using zebrafish and mouse models. She was co-supervised by professors Langyi Chen and Yanmei Liu. She then conducted her postgraduate training at the University of Oxford with Professor Paul Riley, working on differentiation of stem cells into epicardial cells and the development of cell-based assays for high throughput drug screening to promote heart regeneration. With keen interest in diabetes and cell therapies, she joined Professor Timothy Kiefer's lab in 2019 to pursue her postdoctoral training at the University of British Columbia. Dr. Zhao received postdoctoral fellowships from JDRF and Michael Smith Heath Health Research BC and other awards supported by Stem Cell Network, BC Diabetes Research Network, and UBC Outreach Program. And her current research interests in the Kiefer Lab are focused on developing methods to generate functional insulin-producing beta cells from human pluripotent stem cells, designing stem cell-derived islet models to understand normal development and probe pancreas diseases, as well as developing novel strategies to manufacture safe and immune-evasive stem cell islets for diabetes therapies. Thanks so much for joining us. We're very excited to hear your talk.
Thank you. Thank you, Monica, for your kind introduction and inviting me to present at the Sugar Science. So it's my pleasure to be here sharing this story with you. And as Monica mentioned, my talk title today is Stem Cell-Derived Islet Models Generated with a Tunable Differentiation Strategy. And this work is recently published in Nature Communications a couple months ago, and you may find more details discussed in the original publication, but today's presentation, I hope to give you some highlights of the paper. So we all know diabetes is a metabolic disorder characterized by elevated levels of blood glucose and results from the destruction or the dysfunction of insulin-producing pancreatic beta cells. And it's estimated that over 500 million people around the world are now living with diabetes. And this figure is projected to reach 800 million in 2045. Exogenous insulin, whether delivered by injections or pumps, can help improve the glycemic control. But dosing remains a challenge, and we can see the patients often suffer the consequences of a fluctuating blood glucose levels, and they typically develop other complications. And in 2000, Dr. James Shapiro and his colleagues first demonstrated that the islet transplant could effectively provide the freedom from diabetes. However, this procedure also has its limitations, including the shortage of islet donors, poor graft survival, and the need for the chronic immunosuppression. And stem cells may provide an attractive solution to these remaining hurdles. In 2021, the Kiefer and Shapiro groups reported the first results of clinical trials using stem cell-derived pancreatic progenitors contained within microencapsulation devices. And they confirmed the presence of functional beta cells within those retrieved devices, but unfortunately, the cell survival was poor, and thus the insulin secretion, until a follow-up trial where they implanted 2 to 3 times as many cells to achieve the glucose control and reduced the patient's exogenous insulin requirements. And in the meantime, industrial companies such as Vertex and Sonova, their strategy is to implant further differentiated stem cell-derived beta cells. And this way, for the first time, the patients achieved full insulin independence. So demonstrating the promise of stem cell-based islet cell replacement therapy as functional cure for diabetes, but also highlighting the advantages of using more differentiated islet cell types for improved efficacy. Now let's circle back to the process of in vitro bench production of the SC islets. So in 2014, our lab and also other colleagues described a 7-stage protocol to produce the insulin-secreting cells from human pluripotent stem cells. And this is achieved by the timed addition of soluble factors in a stepwise manner through mimicking the embryonic pancreas development in vivo. And specifically, these protocols recapitulate multi-step transition towards definitive endoderm cells, pancreatic progenitor cells, and ultimately the insulin-producing beta cells. And these processes are normally monitored and examined by expression of the cell type specific transcription factors and markers. For example, the FOXA2 and SOX17 are expressed in the definitive endoderm cells, and PDX and NKX6.1 should be turned on in the pancreatic progenitor cells, and functional beta cells normally express insulin, NKX6.1, and MAFE, etc. In addition to these transcriptional changes or transitions, the pancreas development in vivo also involves multiple morphological changes. For example, the pancreatic budding, branching morphogenesis, and establishment of tip trunk domain, where the endocrine cells are specifically developed within the trunk domain, and islet formation, cell remodeling, and architectural reorganization. And all of which they are demonstrated to be important for the establishment of functional islets. And we believe understanding these complex processes of human pancreas morphogenesis may provide new insights into the design of islet cell therapies with optimal function and cellular organization. However, such studies are facing the challenges of limited early stages of human pancreas samples or the lack of complexity in islet organoids generated using current bulk differentiation approaches. So this motivated us to think, can we establish an easily accessible experiment model using the human pluripotent stem cells that could mimic both the transcriptional and morphological changes during the pancreas organogenesis? And we believe the answer is yes. So the big takeaway from our studies is that we find by slightly lowering down the Wnt signaling activation level at the endoderm stage, a budding islet model can be established in the human stem cell cultures, where we can see pancreatic budding, islet formation, as well as the cytoarchitectural reorganization occurs. And this is typically not seen in most conventional bulk type differentiations. And we also propose this budding system could be used as an in vitro model to help understand normal pancreas development and islet morphogenesis, as well as probe disease conditions. So now to show you this budding model from the beginning, we first used an insulin reporter line with a Wnt low protocol. And we noted that in the experiment, only a proportion of the cells is committed to islet cells as indicated by the in-situ fluorescence. But to our surprise, rather than randomly dispersed throughout clusters, these insulin-positive cells were always found to be enriched in local bud-like structures at all stages during the endocrine differentiation. And this islet bud type is very consistent and reproducible as we examined multiple different stem cell lines, including both iPSCs and ESCs. And we then did live cell imaging to track individual clusters during differentiation and observed that actually the appearance of a budding structure always preceded the induction of the insulin-positive cells within them. So we asked the question, what are these initial budding structures and what forms these initial budding structures? We think answering this question will help understand how the islet bud phenotype happens. So the first thought came up into our mind is to examine a panel of early progenitor cell markers. And among them, we find that the cells expressing the transcription factor PDX1 were initially dispersed, and then they seem to cluster together and formed a bud. And this is confirmed by live cell imaging of the cultures with a PDX1 reporter line. So here showing that the initial budding structure is formed by the PDX1 cell clustering. And then the insulin positive cells were gradually induced and restrictively located within the PDX1 enriched bars. So illustrating that the islet bar phenotype is actually rooted on an early pancreatic cell clustering. And indeed, formation of the PDX1 enriched bars appears to create a local favorable niche. And within the niche, the endocrine cells developed in a way similar to the progression normally seen in bulk type differentiations, as shown here. And next, we focused on looking at the infrastructure of these differentiating islet bars. So in addition to the insulin-expressing cells, we also find the islet bars also contained glucagon-expressing alpha cells and somatostatin-expressing delta cells. However, these islet cell types seemed to adopt different organizations at different stages. So the stage 6 bars mainly adopt a mixed organization with the islet cell types intermingled, whereas the stage 7 bars prefer to adopt a core mantle organization with beta cells in the core and alpha cells in the periphery. So to further explore what factors may contribute or could drive such architectural changes, we test the stage-specific additives in the stage 6 and stage 7. And the result shows that the morphogen signals targeting the Notch and BMP signaling, as well as the AXL kinase, and also the antioxidants are able to regulate and pattern the islet bar cytoarchitecture in a synergistic manner. And next, we wanted to explore the possible link between the islet structure and its function. So here we used a microfluidic chip system for simultaneously imaging the calcium activities and also detecting the insulin secretion in the perfusid in individual islet bar. And we here observed that the stage 7 cells had more synchronized calcium responses, but the stage 6 bar had a less synchronized calcium responses upon glucose stimulation. And we also reason that this more synchronized calcium responses is consistent with what we observed in the 6-7 bar, that they have more homotypic basal interactions with the core mantle organization. And so together, we found that the improved cell maturity, as well as this cytoarchitectural reorganization, may explain why the core mantle structure renders our islet bars with better functionality than a mixed one in our stem cell model. In a short summary, by fine-tuning the Wnt signaling activation level during the endoderm stage, we establish a pancreatic cell clustering model for the islet formation and morphogenesis. And this paradigm seems to be supported by like zebrafish and lamprey model animals, where their dorsal bar is formed through convergence of PDX1 positive cells that later develops into a single principal islet. So suggesting that it could be a conserved biological process or mechanism. But regarding to our in vitro human stem cell models, there are still things that we don't understand and requires further investigation. For example, what are these PDX1 negative compartments? So we named the main bodies. Can we examine the transcriptomic profile and determine their cellular identities? And we're also curious to know the mechanism behind of the PDX1 cell body morphogenesis. And we wanted to know if this morphological change is important for the subsequent endocrine cell development. And we also wanted to know, can we use this budding type stem cell-derived islet spheroid system for modeling pancreas diseases in a dish? So first, to tackle the first question, what is the possible cellular identity of the main body cells? So we perform the bulk RNA sequencing on the BARD and main body compartments, because we are able to isolate the intact islet bars from the main body clusters. And this analysis was done by my amazing collaborator, Howard Chen from Dr. Jim Johnson Lab. And he shows that the barred cells and body cells have very distinct transcriptomic profiles. And he also identified thousands of differentially expressed genes between the barred compartments and main bodies. Not surprisingly, the barred cells express higher levels of underquiet lineage, islet cell types, and base cell function markers. But in addition, informed by this bulk RNA-seq data, and then we also validated with immunostaining on selected genes, we found that the main body cells are characterized to express higher levels of pancreatic mesenchymal markers, as well as ductal cell marker and anterior foregut marker, indicating the possible cell types and cellular identity of these main body cells. But the exocrine marker trypsin is not expressed, so ruling out the exocrine identity of the main body cells. After briefly characterizing the identity of the main body cells, next we sought to explore the possible mechanism by which the PDX1 cell body morphogenesis occurs in our stem cell model, and we're curious to know whether it's important for underquiet cell development. So we performed pathway enrichment analysis in order to look for possible candidate pathways that could be involved in the PDX1 cell body morphogenesis. And among them, interestingly, we noted the hippo-YAP mechanical sensitive signaling and also the exon guidance signaling were differentially enriched and specifically upregulated in the main body cells. And then we put a focus on testing the exon guidance cues in this work, as well as also test the signaling pathways related to cell migration as reported in previous literatures, such as the robo-slit signaling, TGF-beta, and canonical Wnt signaling. And interestingly, with our initial screening test showed here, we found only targeting and inhibiting the FB signaling can efficiently interrupt the PDX1 cell clustering or budding. And furthermore, we also tested with more specific FB inhibitors. So we found that perturbation of both FB3 and FB4 signaling can efficiently interrupt the PDX1 cell clustering or budding morphogenesis in a dose-dependent manner. And next, we wanted to know if disruption of the PDX1 cell body morphogenesis could have an impact on subsequent underquiet cell development. So we transiently co-inhibit FB3 and FB4 signaling during the budding stage. And indeed, such perturbation efficiently blocked the PDX1 cell budding, but this also markedly impaired pro-underquiet cell commitment and also the islet cell specification, as well as compromised the insulin secretion compared to the DMSO-treated control. So together, these results emphasize the importance of the PDX1 cell clustering, a process that we identified it requires the FB3 and FB4 signaling in the islet cell development and the base cell function. And the last part of this work we are interested to touch on is that can we use this newly developed budding type stem cell derived islet model to probe pancreas diseases in a dish? So to highlight an example here, we choose the Mitchell-Riley syndrome, which is caused by mutation in the RFX6 gene and characterized by pancreatic hypoplasia and neonatal diabetes in patients. So the RFX6 is a transcription factor known in the maintenance of adult alpha and beta cell function. However, it's not fully understood whether RFX6 affects early pancreas development or morphogenesis. So we aim to address this question in our stem cell models using both differentiation pipelines. And we find that in both cases, knocking out the RFX6 does not impact the commitment of the early endoderm, definitive endoderm cells, but it greatly reduced the induction of PDX1 positive cells. And interestingly, we also noted that the induction of the PDX1 caused by this RFX6 deficiency. So we also find that this reduction caused by RFX6 deficiency switched the bulk type to a budding type differentiation pattern with the observations of islet bud phenotypes. But despite this bud formation, commitment to pro-endocrine cells and islet cell types were greatly impaired, but with the exception that the PPY positive cells were unaffected. And in addition, with the budding pipeline, the PDX1 cells were further decreased in the RFX6 knocking out cultures. And morphologically, these sporadic PDX1 positive cells formed even smaller buds with fewer or no islet cells developed. So together with the both differentiation pipeline, we hope you could appreciate that with our stem cell models, we reviewed an unexplored role of the RFX6 in regulating the pancreatic cell patterning and potentially the early pancreas morphology. And we also demonstrated using this proof of concept examples, we demonstrate the utility of this tunable differentiation system in modeling pancreas diseases such as the mitral Rayleigh syndrome and maybe probing potential mechanisms. So to put them together, we established a budding type stem cell-derived islet spheroid model by fine-tuning the Wnt signaling at the endodome stage. And we also find in an asynchronous differentiation or multicellular environment where there is a considerable PDX1 negative cells, and we found the PDX1 positive cells are able to come together and bud out. And we also identified this process that requires FB3 and FB4 signaling. We further find that the PDX1 cell-enriched budding structures, they may create a local favorable niche, likely resembling the pancreatic buds. And within the niche, the progenitor cells are efficiently committed to islet cells. Lastly, we also touch on, because we can see the cytoarchitectural changes in our stem cell models, and we identify the spatial organization of the islet cell types can to some extent be patterned by morphogen signals, as we identified BMP signaling, Notch, and AXL kinase inhibitors. They can change the architecture, and as well as the antioxidants. And the architecture changes is tightly linked with their functionality. And we also provide proof of concept examples to show the tunable differentiation system can be used for modeling pancreas diseases and probing mechanisms. But we are also aware there are remaining questions and limitations of this study. For example, the degree to which this newly developed body model could mimic the human islet development requires further investigation. And the second, although we use the bulk RNA sequencing to try to probe the transcriptomic profiling of the main body cells, however, the exact cellular identity of them and their exact role in the early islet bud morphogenesis or differentiation is unclear, as well as the crosstalk between the bud and main body, it still requires further investigation. And so with that, yeah, that's pretty much of today. I'd like to thank my mentor, Dr. Tim Kiefer, and my lab members, especially Shenghui and Rob, for their tremendous help. And I also would like to thank my wonderful collaborators for their helpful discussions and valuable input for this project. And very importantly, this work can't be done without the generous support from all these funding resources. And lastly, I'd like to thank Monica again for inviting me to present at the Sugar Science, sharing my research. And thank you everyone for staying with me, and happy to take your questions.
Thank you so much, Jia. That was great. Really interesting new system. And I wanted to open it up for questions. If anyone's here, here's a question. You can unmute Anu.
Yep, that was a nice talk, Jia. I just had two questions. One, in the last slide, you were talking about the limitations. So when you are developing the stem cell derived islet spheroids, you'll have the beta cells. For that, you provide all the signaling, so it can eventually become insulin producing cells. But how do you make sure that it's the alpha cells and delta cells are in place, because they all crosstalk, right? The different cell types in the fine. Yeah. So, so what's your take on that? Like when you compare to the native one, stem cell derived one, like there will be differences in terms of their localization or their cell to cell communications if there are not enough alpha cells, for example.
So if I understand hear your questions clearly, for the cytoarchitectural changes within the islet bars here, so because it's in a differentiating stem cell system, so two things happens. So one is that the cell identity, they're still changing. So we can see the cellular identity changes. For example, at the stage 6 islet bars, we can see some bihormonal cells. So their identity is still not very defined. And then developing to stage 7 islet bars, we can see more monohormonal cells. So here, if we look at the cytoarchitecture, not only the cellular identity that changes and develop, but also their spatial organization changes. But here we are not pretty sure whether those cytoarchitecture change is because of the cell migration. For example, the alpha cells migrate to the periphery, or actually there is a transition of the outer cell, they transition to a favor to a more alpha cell phenotypes. We are still not unsure about this, but we think if we could have a dual report line, for example, the alpha cell and beta cell dual report line, so that and we can track our living cell cultures, and then we can answer this question in future study.
Yes, yeah, thanks. Now the second one question I have is related to RFX transcription factor. Is there any mutation reported in that, or do you know of anything? Can you tell a little bit about that?
You mean the RFX6 reporter line?
Yeah, the particular gene. Why did you study that? Like,
oh, you mean why I chose this gene to? Okay, yeah, this is a very interesting story. Actually, because when we've initially developed the body model, and we found the key point is that if you can induce a considerable amount of the PDX1 negative cells, for example, like in our nature, the pancreas, they are not all, I mean, in an adult pancreas, not all of these pancreas, they express the PDX1 high cells, but only the PDX1 house located eventually in the islet cluster and specifically in the beta cells. So we figured in our body model, the key point is that if we have some proportion of the PDX1 positive cells, and in the meanwhile, we also have the PDX1 negative cell. And though these two compartments, they are at comparable or considerable level, and then the PDX1 positive cells, they can budding out from the negative environment. And then, so we are thinking, if we want to see a body morphogenesis in another model or in a disease model, we hope to find a genes that it can affect PDX1 expression. And then we search the literature, and we actually in our publication, we also probe the PDX1 knocking out using the PDX1 knocking out stem cell lines to probe to mimic and probe the PDX1 now pancreatic agenesis phenotype. But here, by searching literature, especially in the mouse, we found knocking out the RFX6 gene, it can compromise the PDX1 expression. So this, we think it would be perfectly fit with our hypothesis that if in a cell culture, if we're reducing down the proportion of the PDX1 cells, but still have some PDX1 positive cells, and then these stem cell derived cultures, we can see the budding phenotype. So as we can see here. So if we knocking out the RF6 genes and the PDX1 expression, they are lowering down, and then we see the budding morphogenesis.
Okay, thank you.
Yeah, thanks. Thanks for your question.
Great questions. Thank you so much.
Hello, very nice
Hi.
talk. I have a question regarding your budding system in general. I think you mentioned at some point that you think this more recapitulate of zebrafish. And so you are proposing that in humans, the same thing happened in vivo, or this is just another system that you can trying to differentiate beta cell like cells?
This is really great question. And this is also we mentioned it kind of a limitation of our study. It's difficult to say how similar and the degree to which our budding model can mimic the human or mouse the pancreas development. Because we don't have the evidence. But the reason why we think it may mimic the islet development in zebrafish model or the lamprey model is because we find in zebrafish, the islet development is super quick, and especially for the dorsal islet development. And people using the transgenic zebrafish line, they pretty clearly identified during the dorsal pancreatic bud formation. First of all, the PDX1 positive cells, they develop like this, and then they migrate and convergence to form a PDX1 positive dorsal bud. And then this single PDX1 dorsal bud can later develop into a single principal islet that expressing the alpha cells and beta cells. And we think this paradigm is very similar to our in vitro stem cell modeling, as you can show here. So that's why we think what we can say is that maybe our stem cell modeling under the human cell conditions that may mimic a condition like in the zebrafish islet development.
And also along the line, so the budding system is more efficient in generating beta cells, or it just is a different system compared with the traditional system?
That's a great question. Another good question. I didn't show here data, but in the paper we compared the differentiation efficiency of our budding system and the bulk system. And we found they have a comparable system, even though our budding system, they have at early stage, they have a slightly delayed progression. For example, our NKX 6.1 turning on is a little bit delayed, but then after that, they can catching up. And at the end of stage 7, we could have the endocrine cell development and islet cell commitment similar to what we obtained in a bulk type differentiation.
Thank you.
Yeah, thank you for your question.
Great. Okay, thank you, Jia. Thank you very much for introducing us to this very eloquent, a special system. It's really beautiful work, and appreciate your taking the time to walk us through it. We'll look forward to your next paper and talk to you again soon. Thank you.
Thank you.
Great
Thank
day.
you, Monica. Thank you,
Bye-bye.
everyone. Bye.